Hi, my name is Savannah Wiegel, and I am a senior undergraduate student at Hendrix College in Conway, Arkansas. During the summer of 2019, I worked in a genetics research lab under the direction of Dr. Andrea Duina, where I began developing a new system. So I'll begin with a brief overview on genetics. The cell is the most basic unit of life, and if we look inside the nucleus of the cell, this is where we have the DNA, which is a double helix, and genes are also located on this DNA. And so genes are like the blueprint for the cell and the organism. So in order for this information to be used, first the DNA must be copied into mRNA or messenger RNA, and then that process is called transcription, and then the mRNA must be made into protein, which is a process called translation. And these proteins are what we use to do work in the cell. And so a brief overview of transcription. The phases of transcription are initiation, where the DNA opens up and RNA polymerase II binds to the promoter, and then elongation, where RNA polymerase II travels across the coding region of the gene, synthesizing mRNA, and then which is messenger RNA, and then in the final step, which is transcription termination, RNA polymerase II falls off and the mRNA is further processed before translation. And so a model organism is a non-human species used in biological research to understand processes like transcription. And so in our lab, we use Saccharomyces cerevisiae, which is known as brewer's yeast or budding yeast. This is a cost-effective unicellular eukaryote, and it undergoes rapid division called budding, which you can see in this image on the right. And these transcription processes are highly conserved in yeast and can be applied to many other model organisms, and that also includes humans. And so our problem, during the elongation part of transcription, RNA polymerase II encounters DNA binding proteins as it moves across the genes. And these binding proteins typically bind to DNA to carry out specific functions, but they can also bind where they're not supposed to, and now can become obstacles for RNA polymerase II. So that brings us to the question, how does RNA polymerase II overcome these obstacles? And so we developed this hypothesis that RNA polymerase II travels across genes with other proteins whose role it is to remove these obstacles that are in the way of RNA polymerase II. And so to design our experimental system, we are going to place an artificial obstacle on a gene that resembles a DNA binding protein. And so once we have the obstacle there are ways that we can identify that protein that help RNA polymerase remove the obstacle. And so to generate this block, we are using a system called CRISPR-Cas9, which you may have heard of, which it's typically used as a gene editing system, but in this case, we are just using it as an artificial block. And so it would function something like this, where RNA polymerase II moves along the gene and is blocked. And then from here, we are able to look for proteins required for RNA polymerase II to overcome this artificial barrier. And so we performed an assay for cell growth, and the fact that we see less cell growth when deactivated Cas9 is present on the gene is promising. And so this is what we want to see to look for proteins that have that help RNA polymerase II get across that block. And then we did an assay to measure relative deactivated Cas9 binding and saw that it was actually binding compared to our control group. And so our next steps are to make a stronger block, because that block wouldn't suffice for the experiments that we'd like to do. And then from there, we plan to identify proteins that help RNA polymerase II overcome this block. And so the broader relevance of this project, transcription is an essential process for life, and that includes human life. And so there are a lot of health conditions that arise because of defects in transcription, and we are studying a fundamental process of transcription that is highly conserved in yeast and will have applications in human biology as well. And so I'd like to thank Dr. Duina for being such a wonderful mentor and research advisor for this project, as well as my other lab members, Zach Waldrop in the Alan Tackett lab at UAMS for all of their assistance, as well as NSF for supporting us through funding and supporting this project. Thank you so much.